Well, great. Thanks everyone for coming. I'll start by just kind of being a little bit almost like vulnerable in the hopes that this will sort of inspire everyone to do the same because I feel like it's a little bit radical to like even at the Foresight Institute, beautiful setting, we're funded to do this workshop to be like, hey everybody, let's like design molecular 3D printers that bond individual molecules to each other. It still feels radical, even though that's basically the kind of thing that the Foresight Institute was like set up to do in 1986, right? If I understand, my mother was pregnant with me in 1986, so it feels a little bit radical. But what we want to do is try to both be sort of looking 10, 20 years out, but also be really concrete, right? So not sort of say, hey, wouldn't it be great if we had molecular 3D printers? Maybe yes, maybe no. But actually say, well, if we this is the exact chemistry challenge that we need to do to be able to solve some subcomponent of that relative to some specific architecture of how you might do that. Kind of confronting the possibility of just abject failure, right? Like this is what I think this type of workshop is for, is that we're discussing something that it just might completely not work. It might not, in some sense, even be possible. But try to get really concrete about these things despite that. So that's also what we want you to start by doing, by sort of just chatting about each of these architectures together, and then it can come up. I think there's a bunch of other things that will come up in the presentations, and we'll see where this goes. But try to really get concrete about these specific ideas. So what Ben said was reprogrammable tool that can create individually specified covalent bonds at scale. Bunch of definitional questions of sort of exactly what that is. We realize that. I think that there is, like the way that I see this as most like easy to describe to a kind of wide chemistry biology audience is sort of this artificial ribosome idea, which again is the kind of thing that's been talked about since 1986 or earlier, which is that if you think about the ribosome, putting different amino acids in and chemically linking them to each other on the peptide chain, is basically saying, well, I'm either going to put, this amino acid is going to get this covalent bond to the next unit, or this other amino acid is going to get it. That's actually basically choosing which building block gets. And it's not just do I mash them together, it's that they actually get this particular peptide bond is going to be built, for this building block, right, next. So it's covalent bonds, specific ones at specific positions that are being formed by some sort of programmable, molecular machine. In this case, the ribosome. In this case, it's a linear chain between finite-sized building blocks. So you're not asking, can I control every individual atom? You're saying, what can you do with building blocks? But there's still specific bonds between those building blocks. And then if you sort of think, okay, well, this molecular additive manufacturing idea, or the kind of this molecular 3D printer idea is, well, what's the generalization of that to 2D or 3D assemblies where you might have, four corners of a structure, and as opposed to just two bonds, the N and the C terminus of the protein, right? You might have a four potential termini that you could attach things, or you might have 8 if it's a cube or what have you. So there's some sort of an obvious generalization of this ribosome-like system to 2D or 3D, but nobody knows how to design that thing. And so we have some people that are talking about design tools and that kind of cross different, protein engineering, chemistry, various disciplines. We're talking about some things that are scaffolds that could do the positioning. That's sort of the idea. And there's also the question of how do you control such a thing. So the ribosome obviously doing it with RNA. RNA sequences, but you can imagine actuating, which of a choice of different building blocks you put in where using something else. So saying it that way doesn't sound that radical, and various other reasons. I mean, David Forrest had a program at DOE called Molecular Additive Manufacturing that funded a bunch of precursor work that you'll hear on this. So at one level, it doesn't seem very radical. On the other hand, if you ask me, well, what's the specific chemistry that I use and what might go wrong with that chemistry to do this, it feels like everybody's totally at a loss on this. And so this is where we want to be slightly less at a loss on those types of things come 2 days from now. So Ben has gone through and sort of tried to look at what are the different potential high-level approaches. Maybe there's a fifth one that will come up, but we thought it's worth anchoring on these. They're not all exactly the same in terms of what their capabilities would be or what their pitfalls would be, but they all kind of get at this question of what's the sort of 2D or 3D analog of a ribosome-like covalent assembly. So one of these is a printer. And the printer could either be moving the building blocks, or it could just be deprotecting, and then the building blocks actually just go by diffusion to where they're supposed to end up, or it could be directly moving the building blocks. And then there's a breadboard, and the breadboard just moves the building blocks there by some kind of self-assembly type process. Or there's like a solid phase synthesis thing. So if you're synthesizing proteins, without actual ribosomes, peptides, in chemistry, you would do it by solid phase synthesis. And so some of Christian Schaffmeister's work is sort of like solid phase synthesis of more like three-dimensional structures. And then there's actually use a mechanical device like a STM or an AFM to move something unspecified to direct which reactions happen. So those are sort of the high-level ideas. And I thought, again, Eric Drexler is going to come up inspiring all of this in some way. I think with the printer approach is sort of the first one. Like the natural thing that one thinks about is like, I'm going to drag this amino acid, let's say some analog of an amino acid or something maybe a little bit bigger than the amino acid that has, let's say, 4 groups or eight possible linkages. I'm going to drag that in and make the right linkage happen to the right other ones. But it's not necessarily that is what it has to be, right? You could just imagine you only have one chemistry in this entire system, which is a chemistry that deprotects a building block, right? So you imagine these different colored blocks represent the different building blocks. The building blocks, when they're getting added to the structure, they just have to be added to a test tube. There could be trillions of them. They're just diffusing in, and they're going to bind to wherever there's an activated or deprotected site, if that makes sense. So where there's a little red star, whichever building block you add in, so in this one sort of in the middle on the right, you've deprotected 2 sites, and then you're just flowing in the green blocks, right? And the green blocks are just going to go in, and they're going to attach to the deprotected sites, and then they can then in turn be deprotected, right? So in principle, you only need one like position-activated chemistry, which is the deprotection chemistry, and the rest is just like diffusing. Now, I don't know if this is practical or not, right? But maybe we're not really thinking practical like tomorrow, but like, is there a way to design with our collective intelligence this type of thing? But this sort of sequential deprotection with some sort of positioning system, that would be only one chemistry that has to be designed, maybe. So I think that's just an interesting observation. And the other observation about this, right, is that amino acids, maybe that's a little small, but some of the other things like what the spiraligomer bricks that I'm calling them bricks that Christian has worked on. Maybe you can make something that's 2 or 3 nanometers in size, right, between those different, so that the different possible things that you could deprotect, let's say, are some spacing from each other, right, because the underlying building block is some spacing in size and is rigid. So you have rigid building blocks that are let's say a couple nanometers in size, and then does that couple of nanometers correspond to something that you could realistically do with a protein motor or a DNA motor or something else that would position within 2 nanometers of spacing. So that's sort of one idea here, and this is something that I think in David Forrest's program, sort of various pieces of this came up. Maybe DNA origami is a candidate for the underlying kind of gantry that moves around this deprotecting enzyme or whatever it is. So again, so that's one option, is sort of the site activation chemistry. Maybe that can get it down to 1 chemistry or 2 chemistries, I don't know. Another option is directly moving the bricks from one place to another. And this is a 2007 picture from Christian, where schematically these 2 nanometer size chemical bricks that have different kind of functional groups that could be separately deprotected or attached with different types of chemistries, are sort of these 2 nanometer size bricks are sort of shown schematically with these squares and circles linked to each other with bonds that I think in a later talk Christian will explain what those double bonds are meant to be. But it's sort of a rigid little building block overall made of these subunits. And so anyway, so what in 2007 Christian is proposing we actually just directly move the building block from one place to another, and then you can snap into place and have the appropriate bonds at the appropriate positions. So this is sort of the printer architecture. This is like the high-level idea of a printer. And then again, just some of the work. Eric is here, who's first author on this paper. I was sort of trying to prototype, can a DNA origami position something with sort of, 2 or 3 nanometer precision. I'm not sure how precise here. It was less than that in this case. Precise enough to, how precise was it in this case? Yeah. Yeah, so they're more like 10 nanometers. But again, this is the very first instance, very first proof of concept instance. This is sort of roughly 10 nanometers that they're positioning in now 2D, where they can attach different DNA strands with a DNA origami printer inspired kind of gantry type mechanism. So I think what we want to do, can you help me with what the 3 questions are? Is just have people just discuss this, because we want people to actually talk about this so that we don't sort of like lose the thread of this as we get into the talks.
Yeah, so if you could open the working group document, if you haven't done so, again, it's bit.ly/mmnotes. And in the first session, you find architecture number 1, a molecular printer. You have the description, like a short description of it. And here you have discussion prompts, and people are already writing in. Wonderful. So let's take 2 minutes. If you have any notes on what is advantageous about this, what is problematic about this, what open questions do you have about it? And after 2 minutes or so, we give you a few minutes to discuss it with your neighbor, just to make sure that you get talking a little bit and at least conceptually wrapping your head around this somewhat. And there'll be a lot more to be filled in during the presentations, but it's just like a first kind of like getting to know this architecture if you don't know it yet. Okay, wonderful. 2 minutes are starting now, and then I'll give you time for discussing with your neighbor.
Architecture number 2, and then thank you to Chris Wintersinger and William Shee for this figure. And they even have an acronym for pretty concrete. This is not really the full architecture. This is more like a particular concrete version. The acronym is SPANDEX, Scalable Positional Assembly of the Nanoscale via DNA-Based Exhibition.
And order was this designed? Was the acronym first, or was it the demonstration?
Chris?
Turn to William.
Yes.
He's the expert acronym abbreviation.
Yeah, so the general idea came first, and then, like usual, what memorable acronym can be made out of this idea. I think that's
And here we have foldamer primitives, typically 3 to 5 nanometers in size, are tagged with a single-stranded DNA staple strand, and they get assembled into some sort of DNA origami-like structure. Obviously, the architecture could generalize. They could all be made of protein. It could all be made of something else. But here, this is kind of like this, instead of an RNA strand that is basically controlling whether linking together amino acids, you have a 2-dimensional thing like a DNA origami that is controlling some building blocks. Maybe they get assembled, and then maybe you change the pH, or you put in an enzyme, or you put in some chemistry or chemistries that catalyze, let's say the upper right corner of all the building blocks to link to the lower left corner of, the ones right, to the lower left of them or whatever, right? So I said that wrong, but you got the idea, right? You sort of forming the corresponding nearest neighbor chemical linkages that would also sort of ratchet these building blocks that you might have just brought to the right position. You would also ratchet them into the right orientation, right? Because they would only be able to form bonds with the appropriate neighbors. So it's sort of, this is a way that you could get a sort of 2D analog of a templated synthesis of some covalent 2D mesh of molecules. Here, I think maybe these are meant to be like proteins or something that only have certain types of linkages to each other. You could display as side chains, special side chains or something like that. So this is like the general idea. This is not really a printer. It's not really reprogrammable in a dynamic way, but it's programmable by the substrate, which is DNA origami, let's say, which is very programmable. So, so this is sort of another architecture, and we would like you to think about this one too. What are the advantages, disadvantages, sub-problems, etc. Yeah.
Yeah, also, since we didn't take quite 5 minutes for the presentation, if you want to add anything, Will, to it, or someone else who was working on it, feel free to do so.
Is this a good idea?
So I've been thinking about a variant of this. So one of the problems with this can be you need to match your building blocks to the size of the DNA origami breadboard. And so I have a new idea that I'm even more excited about that involves a kind of implosion fabrication, to use the steal the phrase from some of Adam's earlier work, where you self-assemble the building blocks on an expanded grid, kind of exploded view assembly of whatever you want to build, and then you program it to collapse.
How?
Through, I have some figures I could show maybe later, but one is, the first order is you make it on this kind of grid-like structure, and then you, like one of those folding fences. In the first dimension, you basically squeeze in one dimension, so you start from like a square, and then it squeezes into a diamond. And then the second order is pre-programmed rubber bands along the other dimension, that after you remove the scaffold, and you cause those rubber bands to snap, and that will give you contraction in the second dimension. But I can show some figures later.
We're very
That'd be
happy
great,
to have
yeah.
you actually give a talk
Right.
on
This
this
is kind of what we
later.
mean by the architectures thing, right? It doesn't literally have to be DNA strands and protein bricks with different side chains or something. It could be something like that.
Cool. So again, same as with the previous architecture, you can scroll down to architecture number 2, a molecular breadboard, and again you have the discussion prompts. And it would be wonderful if you could fill those in, and then in a second we're going to give you some time to discuss them with your neighbors.
Okay, great. And also, I just want to just say, before we move on to architecture 3, I just want to say a few things about this figure that I realized that I think I didn't quite walk through. So if you look at the blue cylinders, right? So the blue cylinders, I think, and William, tell me this is wrong, but I think the idea of the blue cylinders is that those are like, you can make a 3D DNA origami structure, right? Where the cylinders are all parallel to each other in 3D. And so the cylinders, right, each cylinder, that's like meant to be like 2 nanometers or so in size, right? Because the diameter of the DNA helix is 2 nanometers so in size. So you have this, so the cylinders are sort of like, the breadboard is like these 2 nanometer, circles, all packed in with each other. And then those, the red or white things, the bricks, or here, the bytes that get attached onto the DNA origami pegboards, yeah, they could be proteins or something that have different, several different functional groups, so that they would attach to their appropriate, 6 different neighbors, or something like that. So that's just the idea. The cylinders are meant to be like individual DNA helices, and a DNA origami, which is like something you could actually do. So for this one, this is this sort of solid phase synthesis approach, and I'm sort of realizing I don't have a fantastic way of like pictorializing this, so I'm somewhat tempted to call on Christian and sort of, can you give us a minute of sort of explaining how you have approached bottom-up solid phase synthesis of something that ends up being a 3-dimensional, atomically precise, linked together structure?
Okay, I will talk about this in a couple of minutes there, but we've developed a synthetic chemistry to make these kind of molecules. So we've made 1,200 of those molecules with completely random sequences in the last 2 years, and we could now synthesize millions of them, and we're just gearing up to do that. Then we want to assemble these into multiple segments connected together so that they would fold up, cross-link, and create bricks that are about 2 nanometers to the side, with groups sticking out on the outside that could link up to other bricks and make even larger structures, but all covalently linked together. We are, ready to implement this idea. It is a shovel-ready project.
And this is kind of like, maybe just intuitively, like, if I was to like go into an organic chemistry class and say, well, organic chemists, like, they build giant molecules. Those are precise structures. Like, hasn't people been doing this for 100 years or something? Maybe you can explain what is the enabling thing that...
Giant floppy molecules, and when they're bigger, they tend to be disordered in a fundamental way, disordered or symmetric. These are ordered and asymmetric, and because everything's linked together through pairs of bonds and multiple cross-links, you can program them so you can control where every atom is in 3-dimensional space within the constraints of the structures. I've also been working on software so that we can describe a problem as a sort of a scoring function, and then use high-throughput computing in order to find the shapes of these molecules that will be able to satisfy that scoring function. and I'll describe that.
And maybe you could just say, I'm sorry to put you on the spot with so many things, but maybe you could also say you could say, well, these things, they're these little bricks, they have certain rigid bonds that they can form with other ones. There's a small number of different types of things that they can hang off that could then connect to other bricks. Why don't the bricks just kind of just all mash together in some you can explain the sort of sequential aspect or the solid phase idea.
Yeah, so them just mashing together isn't really a problem, and there are thousands of groups that we can put on these that would be complementary, so that if you have multiple groups that kind of come together at the same time, one of them links up and the other one would, and the other one, if they're properly placed, and you've got the right kind of groups. That would take a little bit more explanation with pictures.
So that was better than I could describe the architecture. Is that part of it is it's sequential, right? So if you're worried about A, bonding with C, you just don't put in A, right? You put A and then you put B, and then B has done its bonding to A, and then you wash out all the B's because the A was attached to a surface, right? And then you put in C, and now C is only going to see the B's, so you can do it sequentially. So part of it is you're restricting by having something attached, and you can wash things off. And then part of it is you have a certain number of different types, and you can put them in some are exposed and some are not exposed.
Right. So examples are, if you have a sulfur and you have a group called a maleimide, those react in a very selective way. If you have a diene and a dienophile, they also react in a very selective way. An amine and an active ester react in a selective way. You can have all three of them together.
All in basically water with some salts, but you basically can cause these reactions to happen. So it's not really, you have a scanning tunneling microscope or something that makes that happen. You just have 4 different types of reactions that are very specific. They can coexist with each other on the same molecule somewhere.
And once you link them, then they're blocked. You are now free to use that scheme again to add something else to it and build up larger and larger structures. I call it kind of a dock and lock approach. You get things close together, they chemically lock, those groups are now hidden away, you can now dock and lock something else to it.
And that idea obviously applies. I think implicit in some of the other architectures of if you put these bricks and then they were getting linked to each other, you could use obviously some of the same types of sort of orthogonal, chemistries, right, that are just reactions that are very specific. There's like this book, Bioconjugate Techniques, that has I don't know how many thousands of different examples of these types of reactions. So this is an example is where you do that with solid phase and very particular building blocks, right, versus having a DNA origami or a printer or something like that. That would be this architecture. Yeah.
Okay, which meaning of solid phase are we using here?
We're using, imagine it's in water. There's the first of these bricks is linked to the surface. And I get to say, well, it's not going to react with brick number 3 until I flow in brick number 2, let it react, and then flow away all the brick 2s and add brick 3. So it's in solution attached to a surface with different things getting flowed on and off is what I mean by solid phase for this one. Yeah, I think what Christian means by solid phase.
Maybe Chris is going to cover this in his talk, but those don't look very water soluble to me.
That one isn't. By putting different groups on them, you can make them. We can control the solubility. Can I also just say I'm tremendously excited about this? You people get me.
Okay, let's start again with 2 minutes, no
Pros and
taking. Feel free to put all of your open questions on there. We will get back to that after the presentations when we actually do the real brainstorming and discussion session. It's mostly just to get your first thoughts onto the doc.
Yeah, it's mostly just to make sure that people are actually are engaging with this, understanding what we're talking. Like if you don't know that explanation of what solid phase is, if you don't understand what that means, talk to your neighbor about that, or go ahead and ask Christian what is he talking about.
Solid phase? It could be all
Yeah, that's a good question. Yeah.
If you have something attached to a solid support, then you can wash away byproducts of reactions and then bring something else in. And if it's in solution, you got to purify it, do some more chemistry, purify it, and that's slower.
So solid phase adds something in your toolkit. If you have really great bricks, I guess you don't need solid phase. Okay, I'm just going to move to the next one. This was our last one for this discussion. Awesome. This one I also feel like is a little bit hard to kind of crystallize in words. There are some words in this section of the document Ben made. But this could either be the thing that's described in this figure, which again sort of seems pretty far out. It's been very theoretical. Ralph Merkle has dozens of patents about this. They also have a funded project in Canada about this, interestingly, per this news release, that has been producing more patents. But here you have a scanning probe microscope. It doesn't just have to be like a piece of metal. It could be molecules attached on the surface in some particular way. Those molecules are then going to go and interact. Typically with this method, this is like more at the level of individual bonds. Like what you're doing is you're exposing some very particular pattern of electron density at the very tip of this tooltip to other molecules on the surface that are going to really drive reactions in some very, this one I think tends to require more like angstrom level precision rather than just building blocks. But then if we go back a little bit, maybe we should also include in this one, this is again from Christian with these spiraligamer bricks. Spiraligomers kind of keep coming up as our go-to example of bricks. But here he has a scanning probe microscope moving bricks around, which are then attaching using these diverse functional groups to each other, plus some amount of spatial control of which brick hits which. So maybe there's a brick-based approach with the scanning probe microscope. You can look up this Canadian banknote nanotechnology and look up their patents, or look up Ralph Merkle's patents if you want to go deeper. Various other literature on this. So far been very theoretical. There have been some quantum chemical calculations of this, whether this will work or not. And there's some people in the room that know a lot more about what are the practicalities. There's a few decades ago, there's a quite deep discussion about can this actually work with surface chemistry. I'm looking at Philip over here. So maybe Philip, I don't know if you want to say anything about this one. Yeah.
I guess all I'd say is, so David and I met nearly 20 years ago at Nottingham. We had a debate. I'm from the University of Nottingham. I'm from the University of Nottingham, which, for those of you who don't know, I guess you know Sherwood Forest, bang in the centre of the UK. So about 20 years ago, David and Josh Hall came over to us, and we had a debate about the future of nanotechnology. These type of designs were discussed. I have also been involved. Ralph was actually a project partner on a fellowship grant I had for a number of years. I will say this, since Idler's incredible pioneering experiment back in 1990, followed up by Crowley's quantum rods in 93, where they slid atoms and molecules across the surface. All wonderful, and I'll talk a little bit about this later on. Beautiful, incredible stuff we can do in 2D. What has there been done in 3D with scanning probes? We're not even at the point where you can build, for example, a 4-atom, 5-atom cluster, because the key thing is controlling the tape chemistry.
Yeah, that's great. Yeah. So I think that this one is there's so many possibilities for what it can be, and there's so many for each of those. There's a bunch of sort of physics questions, but. If you had to take one reference example, I would say what's the latest Ralph Merkle patent, but if you wanted to think about other things, great. And maybe I think your presentation and a couple others will be really the way that we would get into this. Yeah.
I think there's another one directly after mine, which is on surface chemistry, and that's the key issue.
Okay, time for note taking,
All right.
and then Q&A.